Spleen is a part of the immune system. It is an organ in the upper left part of the abdomen, to the left of the stomach. Varies in size and shape between people, but it's commonly fist-shaped, purple, and about 4 inches long. The parts of the spleen. The splenic artery brings blood to the spleen from the heart. The blood leaves the spleen through the splenic vein, which drains into a larger vein, the portal vein, that carries the blood to the liver. The spleen has a covering of fibrous tissue, also called the splenic capsule, that supports its blood vessels and lymphatic vessels. The spleen is made up of two tissues, the white pulp and the red pulp. The white pulp, which is the part of the infection-fighting immune system, produces WBCs called lymphocytes, which in turn produces antibodies, while the red pulp filters the blood, removing unwanted materials. It contains other WBCs called phagocytes that ingest microorganisms. It also monitors the health of the RBCs, destroying those that are abnormal, too old, or damaged to function properly. It also serves as a reservoir for different elements of the blood, especially white blood cells and platelets. The spleen also has a trabecula. It is a small bar rod bundle of fibers or a septal membrane. The spleen has lymph nodes, which is a mass of tissue that contains lymphocytes that filter and clean lymph, while the blood-filled splenic cords and splenic sinuses are found in the red pulp.
Now let's take a closer look at what is happening inside the spleen. Blood passes through the trabecular arteries and into an area enveloped by the periarteriolar lymphoid sheath. Majority of the cells found in this sheath are T cells. B cells can also be found in the sheath. These cells can form nodules as the largest masses of white pulp, and around these nodules are located the marginal zone sinuses. B cells located within the pulse may be activated by a trapped antigen from the blood and form a temporary lymphoid nodule like those of other secondary lymphoid organs. Each central arteriole eventually leaves the white pulp and enters the red pulp, losing its sheath of lymphocytes and branching as several short straight peninsular arterioles that continue as capillaries.
Blood flows inside the red pulp in either open or closed circulation. In closed circulation, blood flows from the central arteriole into capillaries or sinusoids. These sinusoids have unusual elongated endothelial cells called.
So these stage cells have special properties that allow separation of healthy from effete detective and senescent red blood cells. These effete blood cells are then halted and removed from the circulation by the mobilization of nearby macrophages.
In open circulation, blood flows from the peninsular arterioles into open-ended capillaries, dumping blood into the stroma of the splenic cords.
All the blood constituents of the blood must re-enter the vasculature by passing through the narrow slit between the stage cells of sinusoids. Except blood cells are blocked from passing between the stage cells and undergo selective removal by macrophages. The healthy blood cells pass through the sinusoids and proceed towards.
Now let us talk about the activation and release of lymphocytes from the spleen. Antigen in the blood may bind with the B cell receptors. They then undergo endocytosis and are degraded in the endosomes. Peptides from the antigens are presented on the MHC class II molecules of the B cell. A helper T cell then binds this B cell and activates it further with a cytokine, inducing recombination in the immunoglobulin genes and stimulating several cycles of cell proliferation. B lymphocytes also interact with scattered follicular dendritic cells, which have long filamentous processes. The surface of these cells are covered with antibody-antigen complexes bound to receptors for complement proteins and for immunoglobulin Fc regions. This causes B cells to attach, become activated, and aggregate as small primary lymphoid nodules. With the help of adjacent Th cells, these B cells now form a much larger and more prominent secondary lymphoid nodule. These secondary lymphoid nodules can be seen in the earlier diagram and are characterized by a lightly stained germinal center filled with large lymphoblasts. These lymphoblasts undergo immunoglobulin gene recombination, rapid proliferation, and quality control. Growth of activated B cells in germinal centers is exuberant and very rapid, causing naive, non-proliferating B cells to be pushed aside and produce the more darkly stained peripheral mantle. Most of these new specific B lymphocytes differentiate into plasma cells, secreting antibodies that will bind the same epitope recognized by the activated B cell. Some of the newly formed B cells remain as long-lived memory B cells. Formation of long-lived memory lymphocytes is a key feature of adaptive immunity, which allows a very rapid response upon subsequent exposure to the same antigen. These newly formed B lymphocytes are released out of the white pulp and into the bloodstream.